The discovery of MicroRNA by Victor Ambros and Gary Ruvkun, recognized by the 2024 Nobel Prize in Physiology or Medicine, marks a fundamental shift in our understanding of gene regulation. Before this groundbreaking work, the central dogma of molecular biology emphasized gene expression as a relatively straightforward process. DNA is transcribed into messenger RNA mRNA, which is then translated into proteins. However, Ambros and Ruvkun's findings on mRNA revealed a new layer of complexity in how genes are controlled, introducing the concept of post-transcriptional gene regulation. The discovery of the first MicroRNA. The story began in 1993. When Victor Ambros discovered the first MicroRNA, Lin4, in the nematode Caenorhabditis elegans, a model organism often used in genetic research. Ambros found that this small RNA molecule did not encode a protein but instead bound to a complementary sequence on mRNA to inhibit its translation. This suggested that mRNA could silence genes by preventing them from producing their corresponding proteins. Despite the novelty of this discovery, the initial reaction from the scientific community was tepid. The mechanism seemed too peculiar, and many researchers assumed it was a quirk of C. elegans and irrelevant to more complex organisms like humans. Breakthrough: LET7 and conservation across species. The turning point came in 2000 when Gary Ruvkun's research group identified another MicroRNA called LET7, which was not just present in C. elegans but was also highly conserved across a wide range of species, including humans and other animals. The conservation of LET7 across evolutionary boundaries was a strong indicator that MicroRNA was not a genetic oddity but an important and widespread mechanism of gene regulation. This finding generated immense interest and opened up new avenues of research. How MicroRNA works. miRNAs are typically about 22 nucleotides long and function by binding to specific sequences on target mRNAs. This binding either blocks the translation of the mRNA into protein or leads to its degradation, effectively silencing the gene. One of the most remarkable aspects of mRNA is its broad regulatory potential. A single mRNA can regulate multiple genes because mRNAs typically recognize short, conserved sequences in mRNAs. One mRNA can control the expression of numerous genes. A single gene can be regulated by multiple mRNAs. Similarly, one gene might have binding sites for different mRNAs, creating a network of regulation that ensures gene expression is precisely controlled in response to various biological signals. This creates a sophisticated network where mRNAs fine-tune gene expression, allowing cells to respond to environmental stimuli, developmental cues, or stressors. Biological importance of MicroRNAs. Research has revealed that mRNAs are essential in many biological processes. Development: mRNAs play a key role in the timing and regulation of gene expression during embryonic development. They help determine cell fate, guide differentiation, and ensure that organs and tissues form correctly. Cell cycle and apoptosis: mRNAs are involved in controlling cell division and program cell death. Apoptosis. For example, dysregulation of mRNAs can lead to uncontrolled cell growth, which is a hallmark of cancer. Disease: mRNAs are now known to be involved in numerous diseases. Abnormal mRNA expression has been linked to cancer, heart disease, neurodegenerative diseases like Alzheimer's, and immune disorders. For instance, certain mRNAs are overexpressed in tumors, where they promote cancerous growth by silencing tumor suppressor genes. Immune system: mRNAs are critical in modulating the immune response. They can regulate the production of cytokines and other signaling molecules that control inflammation and immune defense mechanisms. Therapeutic implications: the discovery of mRNA has vast implications for medicine, particularly in therapeutics. mRNA-based therapies: researchers are exploring ways to manipulate mRNAs to treat diseases. For example, mRNA mimics can be used to restore the function of mRNAs that are underexpressed in diseases like cancer, while mRNA inhibitors can block overactive mRNAs that promote disease. Diagnostics: MRNA expression patterns can serve as biomarkers for certain diseases. For instance, altered levels of specific MRNAs in blood or tissue samples could help diagnose cancers or predict the progression of diseases like Alzheimer's.